Hello, students, and welcome. Today, we're going to explore two very important parts of the cell. We'll start right at the outer boundary of plant cells and then journey deep inside to find the cell's main control center. First, just look at this image. These are cells from an onion peel. Do you see how they have a very distinct, almost box-like shape? Have you ever wondered what gives them this fixed, rigid structure? Well, that's exactly what we're going to find out. So, here is our plan for today's lesson. We will begin with the plant's protective wall. Then, we will look at a process called plasmolysis. After that, we move to the nucleus, the control center, and understand DNA. We'll also compare two major types of cells, prokaryotes and eukaryotes, and then finish with a quick summary. Okay, first up, let's talk about the plant's protective wall. This is a very special feature that plant cells have. Animal cells, like the ones in our bodies, do not have this. So what is this cell wall? Well, it's a tough, rigid outer layer that you find just outside the plasma membrane in a plant cell. You can think of it like a strong fence or even the wall of a fortress, giving the cell a definite shape and a lot of protection. Now, what makes this wall so strong? The secret ingredient is a substance called cellulose. Cellulose is a complex material, and its job is to provide structural strength. This strength doesn't just help one cell. It helps the entire plant stand upright and tall. The cell wall has several very important jobs. As we said, it provides a fixed shape and strength. It also acts like a shield, protecting the cell from mechanical stress. And here's a crucial one. It prevents the cell from taking in too much water and bursting. It's also fully permeable, meaning it allows water and other substances to pass through freely. Now, let's see what happens when we put a living plant cell into a specific kind of environment, a stressful one. This brings us to a very interesting phenomenon called plasmolysis. So let's imagine an experiment. We take a living plant cell and we place it in some water that is either very salty or very sugary. What do you think is going to happen to the cell? Something quite dramatic happens. Through a process you may have heard of, called osmosis, water will actually move out of the cell. As the cell loses water, all its contents, everything inside the cell wall, will start to shrink and pull away from that strong outer wall. This entire process is what we call plasmolysis. Now, here is a very important point to remember for your exams. Plasmolysis can only happen in living cells. And why is that? It's because osmosis needs a living, functioning, selectively permeable membrane. In a dead cell, this membrane is damaged, so osmosis cannot happen, and therefore, plasmolysis cannot happen either. All right, so we've seen how the cell wall protects the cell from the outside, but what's controlling everything from the inside? For that, we need to travel deeper into the cell, to its control center, or its brain, the nucleus. If you were to look at a cell under a microscope, the nucleus can be hard to spot. To make it visible, we have to use special chemical stains, like an iodine solution or sofrenin. Once we add the stain, the nucleus absorbs it and usually appears as a dark, spherical or oval dot, typically near the center of the cell. The nucleus isn't just a simple blob. It has its own structure. It's surrounded by a double-layered covering that we call the nuclear membrane. This membrane has tiny little holes or pores which allow materials to move in and out. And most importantly, inside the nucleus is where we find the cell's precious genetic material. So, what is this genetic material? Well, it is DNA. You can think of DNA as the blueprint of life. It is the complete instruction manual for building and running the entire cell and, in fact, the entire organism. Now, in a cell that is just going about its daily business and not dividing, this DNA exists as a jumbled, thread-like mass. It looks like a tangled ball of very fine thread. This form is called chromatin. But when the cell gets ready to divide it into two, it needs to be very organized. So this tangled chromatin material condenses and coils up tightly. It forms these thick, visible, rod-shaped structures that we call chromosomes. It's like taking that messy ball of thread and neatly winding it onto spools. And on these chromosomes, we find something called genes. Genes are basically functional segments of DNA. So if you think of the entire DNA molecule as a giant cookbook, then a single gene is like one specific recipe. It contains the instructions for one particular trait, like your hair color or eye color, which you inherit from your parent. Now, the presence of a proper, well-defined nucleus with a membrane is so fundamental to biology, it actually divides all life on Earth into two major groups. This is the great divide between prokaryotic cells and eukaryotic cells. This table shows the key differences very clearly. Let's focus on the most important one, the nuclear region. Eukaryotic cells, which make up plants and animals like us, have a true nucleus, properly enclosed in a nuclear membrane. But prokaryotic cells, like bacteria, don't. Their genetic material just floats around in a poorly defined region called the nucleoid. Also notice that prokaryotes are much smaller, have just a single chromosome, and are missing other complex parts called membrane-bound organelles. Here's a diagram of a prokaryotic cell, like a bacterium. See how there is no distinct circle for a nucleus? The genetic material is just concentrated in that one area, the nucleoid, with no boundary around it. So a common misconception that students have is that bacteria have a nucleus. They do not. They have a nucleoid. All right, that was a detailed journey from the outside of the plant cell to its very core. Let's quickly recap the key takeaways from today's lesson. Please remember these key points. The plant cell wall provides support and protection. Plasmolysis is the shrinkage of cell contents, and it happens only in living cells. The nucleus is the control center, and it contains chromosomes. These chromosomes are made of DNA and carry our genes. And finally, the presence of a true membrane-bound nucleus is what defines a eukaryotic cell. So, I will leave you with this question to think about. How do these two very different components, a simple, strong outer wall and a complex, intelligent inner core, work together to define the unique life of a plant cell? Thank you for joining our lesson today.